Thanks, Richard. It's great to be over in the West again. I haven't been here for quite some time. And it's really interesting in today's program that I'm talking about gene technology, and it seems rather old hat and innocuous compared to some of the things that have just come before here. So I think it's very interesting, some of the developments that are coming on. I was asked today if I could talk about a little bit about how we do gene technology, how we modify plants to have particular characteristics, and just at a very superficial level with the science, so it's not going to be a very heavy scientific presentation, and to talk about some of the traits that are already in agriculture, what their basis is, and some of the things that are coming in the pipeline. And my experience is, as you would realise from Richard, is basically in oilseeds. So the examples that I'm giving here are going to be around oilseeds, but effectively we're giving the same talk with cereals biotechnology as well. So I want to start by the context that we apply plant biotechnology, and really I like to start with this talk around the global this slide around the global challenges, because if we look back at the Earth from a faraway place, it's obviously pretty serene. But in reality, we have some very big issues that are confronting the planet. As we've heard this morning, by 2050, we'll have another 3 billion people compared to what it was in 2010. So we'll have 9 billion people on the planet. And this presents quite a lot of challenge, particularly in the area of food demand. How do we feed those people? How do we produce 70% more food than we produce today? And how do we do that when we've got such limited capacity to increase our land into cultivation, where we have constraints around water, both availability and quality, where nutrient inputs to agriculture are getting more expensive, and where energy is also getting more expensive. And so this really sets a big challenge for us in food security, to improve the productivity of our crops, to improve their security, so make sure they're not affected by diseases, and to improve their nutritional value, because it's not just a matter of calories for eating, you also have to eat nutrients. So we have to make sure that any increased production has got a high nutritional value as well. And I think it's pretty clear now that if you look at the progress we've made with traditional breeding over the last few decades, the incremental improvements that we've been making, 1 to 1.5% per annum in terms of productivity, aren't going to keep up with this increased demand. So we really have to think of some transformational technology steps to get there. I guess the second point I'd make is it's not just about food security. We also have a big problem with materials and energy. So the graph on the bottom there shows you the price of crude oil over the last 150 years, and you can see that long period, very low cost. And based on that, our entire industrial economy has been built around crude oil. And now we've been moving in the last couple of decades into this increasing real price. And have we passed peak oil? Well, we heard this morning the issue around peak oil, so I won't go into that. But at some point we're going to get to peak oil, and we'll see an escalation in prices for crude oil. We know it's a finite, non-renewable resource, so we really have to eventually move society away from a reliance on crude oil for our carbon source. And of course we know that when we use carbon, geocarbon if you like, whether it's petroleum or whether it's coal, we increase the emissions and we start to get the greenhouse effects and climate change impacts. So we have to move away, not just because it's a finite resource, but because of the impact on the atmosphere. Now, we can move to non-carbon sources for a lot of our energy requirements, so solar and wind and hydro, particularly for our stationary energy requirements. But for things like fuels, we still need to have liquid fuels, obviously, for transport, for aviation, for vehicles that can't be electrified. And so we're going to have to move away from geocarbon to bio-based carbon for those fuels in the future. And currently plants are the best option for that. And so for those reasons, I'm very buoyant about the prospects for agriculture in the decades ahead, both from the food perspective with increasing demand and the possibility that we can help support a transition to a bio-based economy. And this is going to lead to a lot of opportunities for farmers, I think, particularly in the next 2 to 3 decades. So this immediately throws up a clash, if you like, and it's commonly called the food versus fuel situation. How do we keep keeping up with the demand for food and also move it to biofuels, for instance, to non-geocarbon fuels? But as I mentioned, and as Annie talked about this morning, it's not just fuel, it's materials. And in fact, fuel is the lowest value option for us. So right now, a biofuel, you just cannot compete with food use of that oil. So at current crude oil prices are equivalent to $500 a tonne, and vegetable oil prices for food get to $1,000 a tonne. So until we get into the area of 150, 160 US dollars a barrel for crude oil, you would not move any oil away from food use into a biofuel use. Materials, on the other hand, are a different matter. You don't really have any other options but to use carbon for materials, and generally they're higher value. So if you look at a petroleum barrel, a barrel of crude oil, about 70% of the volume in that barrel goes to fuel, but it generates about 30% of the value. The other 30% goes to petrochemicals and generates 70% of the value. So in terms of how agriculture positions itself in a bio-based economy, it's really the material sector that we should focus on first. So it has to be food plus fuel plus materials. And really in a constrained resource economy, then that means we have to get more intensified in our crop production systems. So we have to have some step changes in whole system productivity. And that's a whole range of things. I'll talk about the genetics, the breeding side, but it obviously is crop management and agronomy, a whole range of other impacts on whole system productivity. But plant biotechnology is going to play a major role, and it already is. So this is a little bit of a primer on gene technology, and apologies to anyone in the audience who is already very familiar with this, but I just wanted to place it in the context of traditional breeding. Mostly breeders, this is a look at what we call the gene pool, if you like. So most breeding is done just by crossing within the species and selecting for superior genotypes and testing those and eventually releasing varieties. You can do that by interbreeding superior lines. You can do it by inducing mutations using chemical or radiation, working within the gene pool of the crop. In some cases, it's possible to cross crop species to wild relatives, other species, because they're sexually compatible, and a lot of improvement, particularly in the horticulture space, happens in this way, and some of the protection of our cereal crops by disease resistance has happened in this way, the movement of segments of what we call alien chromosomes into the cultivated species. But what genetic engineering opens up is the possibility of sourcing genes from anywhere in nature. So you're no longer restricted by the natural crossing ability of your cultivated species to the organism. There's unlimited access to the genetics of all organisms. And so we can use solutions that nature might have found for particular problems in bacteria, in fungi, in animals, and can potentially apply those into plants. Now, there's been quite an evolution in gene technologies over the last decade or so, and I've listed there some of the capabilities. We can just insert a single gene into a chromosome of the crop plant, say Roundup resistance, Bt, insect resistance. It's possible to insert multiple genes, and I'll give you an example with the DHA synthesis, where we can introduce a whole pathway, 6 or 7 genes. It's becoming possible to put those genes in specific locations within the genome of the plant species, so we can think of having a landing pad, if you like, in a chromosome, so the transgene will always go to the same place. It's been possible to introduce very large sets of genes as mini chromosomes into plants. We can also turn off genes in plants in a specific way, so we can turn it off in this tissue but not in that tissue, through RNAi technologies, which were developed in CSIRO. And a really interesting one for the future, just starting to be applied now, is that we can actually go into the genome and edit the sequence of the genome without introducing transgenic DNA. And this opens up the possibility of having genetically engineered plants which are not regarded as GM in the regulatory sense. And I think we'll see some of that in the future. Well, I think there's obviously a wide variety of opinion around GM technology in the community, and it varies from tremendous potential to help alleviate hunger and supply food for 9 billion people through to this is scary stuff and we should be really worried about it. And perhaps Annie, I'm sure she's still here, but she was kind of throwing down that gauntlet this morning that we should be fearing the gene. And I hope by the end of today you don't fear it as much as you might have at the start. Because it has been a very successful technology, and if you look at the global adoption, these are the latest figures. About 17 million farmers are growing GM crops in 28 countries, about 170 million hectares of biotech crops, if you like, globally. And the interesting point here is about half of those hectares are in developing countries and half in industrial countries. If we just look at the major traits that have been commercialised so far, and you can see that in terms of crops, it's the main crops the big agro-biotech companies are dealing with, and particularly in the US where most of the technology started. So soybean, maize, cotton and canola are the major crops that have GM traits deployed on them. If you look at the penetration by crop, you can see across here that in cotton, about 82% of global cotton production is now GM cotton, predominantly BT-resistant cotton. In soybean, it's 75%, in maize, 32%, and canola, about a quarter of the canola grown in the world is GM canola, herbicide tolerant. And so if you look at the traits, herbicide tolerance, so this would be Roundup Ready and Liberty Link predominantly, way ahead of insect resistance, the BT technologies, but the combination in yellow here is increasing where you have BT and Roundup Ready together. Okay, so how do we make transgenic plants? Firstly, this is just a simple diagram to remind you that DNA, the genes, are contained in the nucleus of the cell, in all cells of the plant, and that we can now, most of our agricultural species, the full genome has been sequenced, so if you like, we know the roadmap of every chromosome in most of our crop plants now, and we know a lot of the functions of the genes because they've been determined in other species, and you can combine that knowledge, if you like. And genes are quite simple, really. They have three regions. They have a control switch, which we call a promoter, that tells the gene when to be expressed. We have a region called the coding region in the middle, which when it's translated will encode the production of a particular protein, and we have a downstream region which is effectively a stop signal. So the coding region produces an RNA molecule, and that RNA molecule is used to synthesize a protein. And that protein can either be a structural protein, like in your hair or your skin, or it can be an enzymatic protein. It can be doing a particular biochemical function. Typical transgene will have two genetic components. One will be the marker gene, which we use as an indirect way of selecting for the gene based on a very clear physical characteristic that we can select for easily, and then the trait gene. And so the trait gene will have that coding region, and it will generally have a specific promoter. So it may not be the same one that comes from that gene in nature, but it's the one which will tell that gene to be expressed in a particular place at a particular time in the recipient plant. So that's very important. And the same components are present in the marker gene. I'd like to show this slide because it shows you just how precise we can be with encoding or directing the transgene's expression within the recipient plant. This shows a transgene which is a reporter gene. So where the gene is expressed, we get a blue dye produced in the cell. And so that blue dye will be produced wherever the promoter tells that gene to be expressed. So in the picture on the left, you can see that dye in some very specific cells during seed development, because that promoter is telling it only to express in those cells. In the picture in the middle, you can see that this is seed in a canola or an Arabidopsis pod, and you can see some are white and some are blue, so you can see this is expressed in seed development and it's segregating in that plant for presence and absence. And the one on the right is where someone has scratched the leaf with a pointy needle, and then the gene has been expressed in response to that. So this is because the reporter gene has a wound-inducible promoter in it. Whenever the cell is wounded, it turns that gene on. So these promoters are useful in responding to pathogen attack, for instance, or pathogen infection or insect attack. So we put genes into plants in one of two ways. On the right is the gene gun way. You can just blast it into a cell on a petri dish and then select for the marker, and the transgene will be in the same cell and grow those plants on. But the normal way is the one on the left, where we introduce it into a bacteria, Agrobacterium tumefaciens, and this is a bacterium which naturally infects plants and inserts its own DNA into the nucleus of the plant. So if we put some DNA in that bacteria, it will insert our DNA into the nucleus and the chromosome of the target plant. And then these plants are taken through a selection system, these cells are taken through a selection system on agar, usually carrying a selectable agent. So we might be selecting for this based on herbicide tolerance or some other chemical tolerance. Only the ones which are transgenic survive. They go on to produce roots and then into a normal glasshouse situation from then on. And we can apply this technique virtually to all crop plants now. This is an old slide, but all of the major crop plants can be transformed using these techniques. So let's have a look at some of the traits that you'd be familiar with. And these are often characterised as first, second and third generation traits. The first wave, second wave and third wave. So in the first wave, they're also known as input traits. These are traits that are really affecting the agronomic performance of a plant. And the classic ones are herbicide tolerance and insect resistance. So Roundup Ready, for instance, how does Roundup Ready work? Well, in every cell there's an enzyme in the middle here called EPSPS, and it's responsible for making a particular class of amino acids that are essential for survival of the cell. So glyphosate works because it interacts with that enzyme and it inactivates it. So if you spray glyphosate on a plant, the herbicide interacts with this, it stops essential amino acid synthesis, the plant dies. So if you supply that plant instead with a version of that enzyme which glyphosate cannot interact with because the molecular structures change slightly, it provides effectively a bypass mechanism. So if you spray the plant, its normal enzyme is interrupted, but it has a backup copy now by the Roundup Ready resistance gene, Roundup resistance gene, and so the plant survives. And I think one point to make about that is that enzyme is present in every cell of your body. It's present in every cell that you consume from plant and animal sources. This is a very innocuous technology, although a lot is claimed about not knowing anything about the long-term effects. This is a very basic gene that's present in all food we already consume. Bt-resistant cotton. In this case, the plants are equipped with an insecticidal protein that actually comes from the bacteria Bacillus thuringiensis, which has been used in organic production for a long time. Now, you might know it as dipel. And dipel works by the bacteria producing a protein which attacks the gut of the Lepidopteran pests. And so all that's happened in this development is to take the gene for that protein and put it into the plants. So the plants are making their own insecticide, if you like. When they're chewed on by the grubs, the protein is ingested and attacks the gut and they die. So you can see in the picture below, on the left, a Bt cotton leaf which has been completely immune to attack. On the right, a control leaf which has been decimated by the grubs. So these traits have already given a lot of benefit to agriculture. In the case of herbicide tolerance, it's enabled us to move to broader spectrum and low residual herbicides, reduced applications compared to using multiple selective herbicides, and been very instrumental in the adoption of no-till system, minimum tillage systems in farming, conservation farming. In the case of the insect resistance, if you look at a crop like cotton, which used to need about 17 sprays to keep the insects away, spraying is very much reduced in BT crops. And so it's been estimated that about 500,000 tonnes of active ingredient of insecticides has been saved since 1996, saved and released in the environment due to this technology. And of course, there are also the saved lives from reduced insecticide exposures, particularly in developing countries. So these already have substantial benefits. Coming behind those traits which are already in the market, there'll be traits for disease resistance, so more durable resistance to pathogen attack or specific pathogens. A really interesting one here is in grapes. Powdery mildew or mildews are a major disease of grapes, and I think about 70% of the agrochemicals used in Europe are applied to grape crops to prevent powdery mildew. And the gene is available for resistance to powdery mildew and could be introduced into grapes today and prevent that use of agrochemicals. And there's also been work done in the horticultural area on reducing post-harvest spoilage, so we can genetically modify the plant so that you don't get the browning when you cut the surface of the fruit. Further down the track, things that are still being worked on, if you like, in the lab, are ways of increasing the yield potential of crops. For instance, you can see on the bottom there a typical hybrid corn variety, 2 parents on either side, and the hybrid of those 2 parents has got very much increased yield. Not all crops have hybrid varieties, so wheat, for instance, we don't have hybrid wheats. But if we could develop hybrid systems in those crops, we have the potential to increase the yield substantially in the same way that's been very successful in corn. There is also the potential to develop pure breeding hybrids. So once you have a hybrid variety, it could keep that performance in perpetuity and be propagated by seed rather than going back to the seed supplier and getting new hybrid seed each year, a process called apomixis. And we have some research in CSIRO trying to develop apomictic breeding systems in crop plants, particularly in wheat. Further down the track, I think improved photosynthesis. Wheat is known as a C3 plant. It has a particular biochemistry which is, in terms of its photosynthesis, which is not as efficient as some of the other grass plants. If you think of bamboo, for instance, and sugarcane, enormous biomass crops that are actually C4 plants. They have a different pathway for photosynthesis. So there's a lot of interest to see whether we could bring that pathway or engineer our grain crops to have that pathway for photosynthesis, to see if we could take it up another level and get into very high biomass cereal crops. And also work to increase the input use efficiency for water and nutrients by both conventional and gene technology routes. And stress tolerance. So the bottom picture at the bottom right, if you can see it, is a barley seedling in an acid soil. So it's very sensitive to aluminium, but you can select introduced genes for aluminium tolerance, which enables that plant to behave quite normally in that environment. So they're the input traits. What about the second wave of technology, the output traits, where we actually modify the product quality? And probably the one you're most familiar with here would be golden rice, where this has been going for some time now, where it's been possible to increase the level of vitamin A, beta-carotene in rice by a factor of about 20. And so you can see at the bottom right here, the golden rice that has the carotene colour, the yellow at the back, compared to the normal rice. And this has got a tremendous potential to alleviate vitamin A deficiency, particularly in the developing world. Other opportunities here, we can consider removing undesirable components from the seed. So imagine, for instance, taking an allergen out of peanuts would be a good thing to do, modifying the problems with gluten in wheat so that celiacs could potentially safely eat wheat, and removing saturated fats from oils is one that we've done quite a bit of work on at CSIRO. And then there's the possibility to add in nutrients as well. It's called biofortification, and I'll give you an example of that where we've introduced the fish oil fatty acids into canola. So EPA and DHA, I had a table talking around lunchtime, not exactly sure what they were saying about it, but fish oil supplements, I think this is a nutrient which probably has the widest knowledge in the community in terms of beneficial effects. It's an incredibly important nutrient. The long-chain fatty acids effectively come from marine sources, mainly from fish, and we need them in every cell of our body, and we particularly need them during fetal development and infant development for brain function and for retinal development. They also have very positive effects in a whole range of conditions, including reducing cardiovascular risk, positive effects on Alzheimer's and cognitive and mood issues. And it's pretty clear that almost everyone gets too little of these fatty acids. We only consume about 20% of what the recommended daily intake is. And if everybody was to move to the recommended daily intake, we just wouldn't have enough fish. We already know that fish stocks around the world are under threat, collapse in many cases. Relying on aquaculture is really an unsustainable approach because currently you feed fish to aquaculture systems. And so we really need to develop some safe, affordable and sustainable sources of these nutrients. And plants offer the best opportunity for us to do this. If you look at the cost of production, for instance, if we were to get 100 grams of long-chain polyunsaturates, EPA and DHA, the fish fatty acids, if we get that through fish, it'll cost us about $75. If we get it just as a capsule, it'll cost you somewhere around $40. If we produce it in plant, it can be produced for around $4. So there's a tremendous value proposition there compared to the current sources. So these are the 5 fatty acids which are present in canola and our other oilseeds. No plants produce EPA and DHA. They only come, no terrestrial plants, they only come from marine plants, the microalgae in the oceans. So microalgae have this additional pathway of converting 18-3 through to a 22-carbon and 6-double-bond fatty acid DHA. And so that takes 5 enzymes for the cell to do that, and fish get their DHA and EPA by eating microalgae. So fish don't make these, they consume them and they move up the food chain and then we eat the fish. So back in 2005, from the Food Futures flagship in CSIRO, we were the first to demonstrate the production of DHA in terrestrial plants by introducing the genes of microalgae for this pathway, all 5 of them, into the plants, and we've got around 2% EPA and 1% DHA. And then in 2012, we took that technology further, and we've now produced up to 15% DHA in the seed oil of Arabidopsis, and are now transferring that technology into canola. So those levels, 15%, that's equivalent to the composition of bulk fish oil. So we're already in those plants producing the equivalent of fish oil. And the calculation is that you could replace 10,000 fish, if you like, in terms of DHA supply with 1 hectare of canola, and that is probably the first output trait that will be commercialised in Australia, and it's currently under development with CSIRO, GRDC and UC. So we'll see how that goes in future years. In principle, the way we currently get DHA, algae make it, fish eat it, we eat the fish, but by putting it into canola we open up the possibility of that oil going into the food supply in a whole range of ways, and the nutrient being there in the background in the diet. So it's a public health type approach, if you like. Okay, so the third wave. Well, the third wave is really about producing industrial traits in plants. So this is about the biomaterials, bioenergy space that I talked about earlier. And I'll give you one example of this, and it comes from our own work. CSIRO and GRDC have been investing in a project called crop biofactories now for a number of years, and we're working with safflower as the crop that was chosen by the GRDC in consultation with growers as a crop that was a small crop in Australia, could become a dedicated industrial crop platform without impacting our food supply. And we've been able to develop just recently a line of safflower which has 95% of one particular fatty acid. It's oleic acid. You see the peak in the middle, if you can see it on the charts there. 95% of that fatty acid. This is by far the most pure plant oil for a single fatty acid. And this really gets us into the chemical space, the petrochemical replacement space, and should have quite a premium in value over the other oils. And that fatty acid would be used in a number of ways, either in industrial lubricants or as a feedstock. You can chop that molecule in half in the middle, make 2 9-carbon monomers which go into plastic production. So this is a raw material for bioplastics. So I think that's really going to become a bit of an icebreaker for GM industrial oilseeds, and we're about to do our first field trial of that this year. Okay, so that's the type of thing we can do by GM, and I wanted to focus on the bio-based industrial side. We've set ourselves a bit of a challenge because obviously we have to produce enough oil to meet our food requirements and to have some left over to do industrial replacements with. It won't be acceptable to produce industrial products if we can't meet our food needs. So to do that, we really have to ramp up the production of plant oils quite significantly from where they are now. And so we've looked at whether we could conceivably produce treble the global plant oil production by 2050 so that we could meet the growth in food and replace 40% of petroleum products using renewable plant oils. So this is the current plant oil production globally, about 140 million tonnes, with a population of around 6 billion at the turn of the century. The increased food demand just with the population growth over the next 3 or 4 decades would take that up to close to 200 million tonne requirement. But it's not just population that's increasing, per capita consumption is still increasing. And so you can see quite different per capita consumption across those range of countries. Clearly, at the bottom end of that spectrum, there's a need to increase it to satisfactory levels. At the top end, there's an urgent need to reduce it to healthy levels. And it seems like that reduction in developing societies is happening a lot slower than the increase in, the reduction in developed societies is happening slower than the increase in developing nations. So I think the per capita consumption globally is going to go up as well. So if we add something for that, we're getting up into the 220, 225 range and a population of 9 billion people. If we want to replace 40% of petrochemical products with plant-based products, we need to get to that level. So effectively you can see that from 2010 to 2050, we would need to treble plant oil production. We've trebled plant oil production previously, since 1985. It took us 25 years to do that. But we were doing that in a situation where we had expanded acreages, where we had increased yields, and now that's going to be a lot harder in the years ahead. So here's 4 ways that we think we could contribute to increasing plant oils. Some people are looking at new crops for marginal land. I think that's not such a good idea for Australia. Our marginal land is already in production, some people would say, so the opportunities to expand our production base is fairly limited. Algal oil production systems are really promising, and that's one way of using our saltwater resources rather than our freshwater resources, but it's going to be some time before the cost of production comes down to make them viable in an economic sense, and to address scalability challenges. But still, I think we need to work on those, and hopefully we can bring those online eventually. The 2 that we're focused on are increasing oil content in our oilseed crops and trying to synthesise oil in tissues other than seeds, particularly in leaves. So if you just look at how we travelled over the past 20 years, we doubled the area sown to oil crops globally, so that's obviously a big factor. we had about a 40 to 50% improvement in crop productivity, so that 1 to 2% per annum that is typical in breeding programs. But we made virtually no changes to the oil content in our seeds. So there's a very large untapped potential. Here's some of our major oil seeds. The best is sunflower and rapeseed at 48% oil, peanut as well, and then quite a few of the other major crops, much lower oil contents, because they're not traditionally oil seeds, particularly, say, cottonseed and lupin I put on here at 8% is not an oil seed at all. But in nature, you can get 60% oil, and sesame has 60% oil. Macadamia has 70% oil. So there is potential to move some of these plants to very high oil production. And so, if you can calculate there the untapped potential, if we could engineer those plants to have the same oil content as some of the plants which already do have high oil contents. And I put lupin up there, particularly because I'm in the West, and we're very interested in this. Lupin obviously is a crop that's been valuable in Western Australia, but has kind of fallen out of favour. It's a very low value in terms of its price, and I don't know what that means. Did that come up? No, sorry. I've got something on my screen here. Yep, that's okay. It has 8% oil, and so we obviously don't crush it for oil. 8% is about the cost that it would take to crush a seed, so there's no net gain there. But what would happen if we could move it to 45% oil, say canola levels? It's an entirely different value proposition then. You would see that the value of the seed go up from around $250 a tonne on this calculation up to $490, so potentially doubling the value of lupin seed. If you could combine that with herbicide tolerance to give you a bit more flexibility in the cropping system there, this now becomes a much more viable option, an opportunity to bring back in a nitrogen-fixing leguminous break crop into your farming systems here. So we are starting some work to see whether this is feasible, whether we can raise contents of oil in lupin seeds. And the model here would be peanut. Peanut is a grain legume, 45%, 48% oil. Soybean is a grain legume, 20%. So some wild lupin species, 20%. So narrow leaf and white lupin, there's the potential for us to change the use of that crop and increase the value proposition to growers. I just want to finish up with one recent development which is, I think, going to be a real game changer. Coming out of some of our really basic research into how plants synthesize oils, we developed a system of testing these pathways in leaves as a way of quickly testing the addition of a gene. You express it in a leaf transiently, just temporarily in the leaf, and you look at what happens. And we found through that process that we were actually able to get leaves to synthesize and accumulate significant amounts of oil. And it involves a technology we call push, pull and protect. So simply, the push is you get the plant to make more fatty acids. The pull is you get the plant to put those fatty acids on a triglyceride, an oil molecule. And the protect is you get it to store that in a protected oil body so that it's not broken down by the leaf. And so leaves normally do produce some oils on a daily basis, but they break them down the next day. So they store a bit of energy, they break it down the next day. We've intercepted that and made them accumulate oil. And we found levels up to around 17% of triglycerides in tobacco leaf. And the picture on the bottom left, you can see that the white panel is a control leaf which doesn't have these pathways expressed and is not showing any oil. And the bottom left figure is the transgenic leaf, which is expressing oil throughout the section there. That's the section across the leaf with the midrib down the middle. And just to show you at a higher magnification, you can clearly see the oil present in these leaves. This opens up a whole new potential. If we can produce 17% oil in a biomass crop, that would mean that if you produce that in, say, sugarcane or miscanthus, something which is a dedicated bioenergy crop, you would have the same oil production as oil palm plantation, about 6 tonnes per hectare. So this is really quite a significant development, and we're now trying to explore what are the limits that we can push this to, what are the potential production platforms that it could be deployed on across across. It could be a whole range of options here, dedicated crops, or perhaps we have it expressed in the biomass of a cereal crop, so that at the end of the season you get your grain and then you have your biomass, maybe going into a biorefinery for energy and chemicals, but first with an oil extraction and a significant value proposition up front. So I think that's that's the latest thing from our lab anyway, and other labs around the world are working on similar technologies there. So just to finish up, a prospective technology path for trebling oil production over the next few decades from where we are today. Can we go from 140 to, say, 420? The first gain will be through productivity gains. That's a continuation of breeding efforts that are going on now. Let's say 1% per annum. We might be able to increase by 70 million tonnes. Maximising seed oil content, so I showed you the untapped potential there. If we can make some significant inroads there, maybe another 110 million tonnes. And then finally, the new plant oil sources is probably the same sort of magnitude. And so I think the prospects are there for using biotechnology to treble plant oil production over a number of decades, and that will allow us to keep pace with the food needs of 9 billion people eventually, and also have a surplus available to use for industrial use to help us replace petrochemicals and move away from petroleum and geocarbon sources in our industrial society. And I'd like to leave it there just by, I think they might come up in questions, by thanking the participants, the researchers in our labs in Canberra who have contributed some of the data that I've included in the slides there. Thank you.
Thank you, Allan, for a very interesting talk. Does anybody have any questions?
I'll ask it that. I just want to ask the obvious question. Why do we fear GM so much? Is it lack of knowledge, or is there a real concern there?
I think those who are opposed to gene technology or uncertain about gene technology have done a very good job of portraying that as a fear to the general public. There are a lot of reasons that people may oppose gene technology. I think the one which is most often used in terms of communicating those fears to society is the safety issue. Yet that's the one which I would be least concerned about, because we have a very science-based regulatory system. It takes an enormous investment to get something through a gene technology, a strategy through to the market. That's why only what I call the killer apps have got to market so far, and with the deep pockets of big companies. So I think it's highly unlikely that the technology will get through that would not be safe. The real root opposition has been, I believe anyway, the concern about the multinational dominance of the technology. And I hope that over time this will become much more democratised as a technology, particularly when maybe some of the regulatory hurdles that eventually come down and some of the trade barriers are dealt with in a more harmonised and better way. So I think they've done a pretty good job. If you're an opponent of gene technology, it's easy to make people scared about it. And probably there's been a bit of a vacuum on the other side around the true situation.
I'm Kevin from the experts. I was just wondering about a frost-resistant gene for wheat and barley.
What's the actual question?
Oh, just because there's millions of tonnes lost every year in WA from frost.
Oh, sorry, frost resistance. Sorry,
Yeah.
it's the glufosinate resistance. I think the frost resistance is an interesting one. I think probably the timing of flowering might be amenable to change there, and I guess we don't know what's going to happen over time with the change in the temperature profile because of global warming. But a specific resistance to frost, like a physiological resistance to it, regardless of the timing of the event, I'm not aware of anything that's in the pipeline at the moment. But I think there's a lot of effort in all of the stress resistances, so maybe there's something out there I'm not aware of.
Any other questions?
Yeah, just one question, George. Yeah, I've got two actually. Just a couple of questions.
I'll just give you one answer.
I was wondering if there's any studies of biofuel. There's the amount of fuel used to plant the crop, spray it, harvest it, transport it, process it. Is there any studies done to see how much is actually left to go back into resupplying for petrols and stuff after we've used it?
In terms of the net energy gain?
Yeah, like is it like viable to do it? We're not just going to small margin. We end up getting a side.
There are net energy gains, but the viability question is really price related more than it is to that, because right now there is no driver to produce biodiesel in a crop plant where you can, that's an oil substitute, right? So take food oil and put it into diesel engines at half the price. There's no bidding away from the food market. The real issue comes when crude oil price reaches, say, $160, $170 a barrel, parity price with food oil. We know that any food oil can go into a diesel application. But most of our food oils are of the composition suitable for diesel. And so at that point, the market bids it to wherever the price is. And that's where it becomes a real issue for society. Do we want to get into a situation where industry will bid vegetable oil to industrial use instead of to food? And I think we might see at that point in time an intervention. Similar to what we saw at the start of the process, which was a positive intervention by subsidy on biodiesel in countries like Brazil and Austria. So long term, I don't see the value being there. If we can make oils which have high value components that increase the value package, the return to growers, and then after you take those high value components out, you have a residue which may go for energy and contribute something to the value proposition. It's commonly called attacking the barrel in petrochemical terms, emulating what the petrochemical industry does. The fuel is the leftover that you can afford to sell at a lower price because you have a very high value component.
And what about with, say if you get a frost tolerant variety and drought tolerant varieties and everyone in the world grows a perfect crop? Well, at the moment, if America have a good year, if the Saudis have a good year, Black Sea has a good year, our prices drop really bad. So if everyone in the world has a perfect year, what happens to our prices? Because there's people in the world starving now that can't afford to buy our grain.
Yeah.
So that's probably it.
Well, I think you have to say then, if you follow that through to the conclusion I think you're trying to draw is we shouldn't do this because it will put us in a worse position. It will put you in, at worst, an equivalent position at a higher production level. And I don't think the reason most people are farmers, I know we've heard a bit about lifestyle choices today, but I'd be really surprised from my contact with agriculture if you're only in this business for lifestyle. I think agriculture serves a major purpose for society. And it's a motivator for farmers, I think, to be both meeting that need of society and making a living and having a lifestyle. So I would argue you should move to as high a production platform as you can, because less people will be hungry and we will have less carbon coming out of the ground to meet our industrial needs. We may be in no better competitive position vis-a-vis other producers, but the world will be a better place.
Okay, on that note, we'll leave it there. I'd like to thank Allan for his presentation.